According to data collected by the Centers for Disease Control and Prevention, there were 979 reported illnesses and 73 hospitalizations between 2007 and 2012 from one source alone: raw milk. Milk is one of the many dairy products we consume on a daily basis. Before it can be consumed, however, it must be decontaminated through a process called pasteurization. I'm Rikka Abano, and during this presentation, I will explain what pasteurization is, where it came from, and its uses today. Afterwards, Ryan will describe the different ways milk can be pasteurized, and finally, Caitlin and Alex will further discuss and demonstrate the underlying chemistry of pasteurization. Pasteurization is a process that uses heat to destroy pathogens in foods. These contaminants can come from many sources, such as soil, dirty water, or sores on the animal being milked. As Louis Pasteur discovered over 150 years ago, these pathogens can cause food to spoil and can cause illnesses like Salmonella, E. coli, Listeria, and Brucella. The prevention of these diseases can be accredited to discoveries made by Louis Pasteur in the mid-1800s. Pasteur was a French biologist and chemist known for life-saving breakthroughs in areas such as vaccination, fermentation, and pasteurization. Through his research conducted on the fermentation of beet juice to make alcohol, Pasteur came across a microorganism called yeast as responsible for the ethanol present in wines and beers. When left untreated, however, yeast will turn into Mycoderma aceti and will cause wine to spoil. Like previous scientists such as Nicholas Appert, the man who first developed food preservation through canning, Pasteur found that high temperatures can be used to kill microbes that can cause food to spoil and others that carry illnesses. While this discovery was made in 1856, pasteurization was not commercially used until the late 1800s. Today, pasteurization can be applied to many types of foods, including syrups, juices, nuts, and dairy products. When producing products such as yogurt and cheese, manufacturers need to monitor the temperature at which the dairy is pasteurized and the pH of the milk. As Caitlin will discuss later, certain pasteurization temperatures can denature proteins and cause flavors and textures that are undesired by consumers. According to a study conducted by Florencia Frau, Graciela Fonte Valdez, and Nora Pese at the National University of Santiago del Estero, an underlying cause of the change in texture is the presence of hydrophilic whey proteins that allow higher moisture retentions in the cheese. On the other hand, acidity measurements can be used to control the flavor and determine when a batch of a product is ready. In yogurt specifically, the standard minimum pH in the US is 4.4. In order to manufacture dairy products that have the correct consistency and flavor, temperature and acidity should be monitored closely. Yogurts rely on vat pasteurization with lower temperatures and longer pasteurization times to achieve its signature body, texture, and taste. Pasteurization temperatures are dependent on the different types used, as Ryan will now discuss.
Vat pasteurization, as you may have guessed from the name, is a form of pasteurization which uses a large vat to heat the products. It is a relatively light pasteurization when compared to the other ones we will discuss, but the products must be kept above 145 degrees Fahrenheit for at least 30 minutes and then immediately cooled to 39 degrees Fahrenheit. Because it is such a gentle form of pasteurization, it is often used for cheeses, yogurt, and most importantly, ice cream. There are various other types of pasteurization, and we certainly won't be able to cover them all in this presentation. Here are a few of them that we will discuss before explaining the chemistry involved with these processes. Some other common forms of pasteurization include high-temperature short-time pasteurization, ultra-pasteurization, and ultraviolet pasteurization. High-temperature short-time pasteurization, or HTST for short, is a more intense pasteurization than vat pasteurization because the products must be kept at 161 degrees Fahrenheit for 16 seconds. This form of pasteurization is often used for milk, juices, beer, and even almonds. Often, products that undergo this form of pasteurization require refrigeration immediately afterward. Ultrapasteurization is a much faster but much more intense form of pasteurization than the previous two forms. It requires products to be heated up to 280 degrees Fahrenheit for just 2 seconds. This form of pasteurization is used for a lot of things like fruit juices, cream, soy milk, yogurt, wine, soups, honey, and even stews. Pasteurizing these products can really help to increase their shelf lives. This process isn't as common with milk because it can cause an unfavorable Maillard reaction to occur and make the milk brown. We will discuss that in more detail later on. Ultraviolet pasteurization is a very unique process which uses ultraviolet radiation, most commonly between 100 and 280 nanometers, to kill unwanted diseases and bacteria. As we've discussed in class, heat can be used to denature certain proteins, bacteria, cells, etc. And that is essentially what most forms of pasteurization do. They heat up the product enough to kill any undesired bacteria or disease, but hopefully not enough to damage the product. Ultraviolet pasteurization, on the other hand, uses ultraviolet waves to kill off unwanted forms of disease and bacteria. It hasn't been tested and authorized for many dairy products, but can be used to pasteurize things like vegetables, fruits, meats, and grains. In high school, I actually worked at an apple orchard, and we used ultraviolet pasteurization to pasteurize our apple cider. This is just one of many unique forms of pasteurization that are out there. Now that we understand the different types of pasteurization and their differences, we can discuss how the different enzymes in milk can be affected by pasteurization, as well as what happens if it receives too much or too little pasteurization.
There are 4 important kinds of enzymes in milk which contribute to milk's pasteurization. These are lipoprotein lipase, plasmin, alkaline phosphatase, and lactoperoxidase. The first type of enzyme is lipoprotein lipase, which is a type of lipase or enzyme that degrades fats. If lipoprotein lipase happens to be agitated during processing, then it comes into contact with the fats in milk, degrading them. This would cause a change in the flavor of the milk. Pasteurization inactivates lipoprotein lipase, which increases the shelf life of the milk. Plasmin is the second type of enzyme in milk. It is a type of protease, which are enzymes that degrade proteins. If plasmin is allowed to degrade the proteins in milk, it causes the milk to have a bitter flavor and different texture. This is why plasmin is used to manufacture cheese. The next enzyme in milk is alkaline phosphatase, a heat-sensitive enzyme. Since it is heat-sensitive, alkaline phosphatase is used as a pasteurization indicator, because if the milk is pasteurized properly, then alkaline phosphatase becomes inactivated. The final kind of milk enzyme is a heat-stable enzyme called lactoperoxidase. When lactoperoxidase is combined with hydrogen peroxide and thiocyanate, it stops the microorganisms in milk that cause disease. Since neither hydrogen peroxide or thiocyanate is present in milk, they must be added to get this property. If milk were to be exposed to high temperatures for longer than the normal period of time, then it will undergo the Maillard reaction, a reaction between an amino acid or a protein and a reducing sugar. The result of the Maillard reaction for milk is browning. The browning of the product happens at the end stages of the Maillard reaction, when enough water has disappeared from the product. In addition to browning, the Maillard reaction can also change the flavor of the product. Some products that take advantage of the flavor-changing quality of the Maillard reaction are beer, whiskey, creme brulee, toasted bread, and meat cooked at high temperature, like barbecue. Here we have the basic process of the Maillard reaction in milk. First, we start out with the sugar and amino acid. Then, when we subject the sugar and amino acid to high temperatures, we get a Schiff base. When the Schiff base is made, it quickly reacts to form an amidori adduct. The amidori adduct then undergoes dehydration and fragmentation to give products like HMF and hydrovaldehyde. Then, when these products undergo aldol condensation, we are left with melanoidins. These melanoidins are responsible for the brown color that occurs in the milk, and the change in flavor and smell as a result of the Maillard reaction.
So today we are going to be discussing the Maillard reaction and showing how it turns milk brown after it's been heated up. So as it heats up, the proteins in the milk are going to be reacting and causing the brown color and a change in odor. definitely see some right there. That milk has turned brown. It has undergone the Maillard reaction. We're trying to get more of it too. As it cools down here, you can see it starting to congeal. I had to stop it early because it was going to boil over, but you can still see that area on the side there, that the Maillard reaction did take place with when there was a lot less moisture in the area where the milk was. So there you have it. That is an example of the Maillard reaction in milk.
In this presentation, we covered the different types of pasteurization, the enzymes in milk, and the Maillard reaction. We discussed 4 different types of pasteurization, including vat, high temperature short time, ultra, and ultraviolet. We then focused specifically on milk and the enzymes in milk. We talked about 4 different types of enzymes, 2 of which are deactivated by the pasteurization process. Finally, we reached the Maillard reaction, in which proteins in food are combined with sugar and heat to brown the food through a complicated multi-step chemical reaction. This has been a pasteurization presentation. Thank you for watching.